All right, everybody should remember how our game is played from yesterday here in the room. If you have questions here in the room, feel free to come up to the microphones. For those of you online, if you have not played the panel game before, the way it works is hopefully you've been typing in your question into that Q&A pod in the lower right-hand corner. If not, now would be a good time to do so. If you have a question for a specific person on the panel, please preface your question with that person's name. Let's go ahead and get started with microphone number 2 here in the room. Step up nice and close for me, please. Oh, sorry, that may not have got powered on. There you go.
Hi, good morning. Question for Mr. Vasudeva and Nguyen. First, intensely appreciated the Captain America references. Really cool. This is around the process of counting the deficiencies. It's kind of a long question. If you were to exceed the number of minor deficiencies or items, let's say you had a number like 13, and after a request for reconciliation, it is determined that you're actually at around 9, so you have less deficiencies than are necessary to trigger an RTR. Does that mean a de facto acceptance of the file, or does it trigger another round of file review in order to, before it is officially accepted?
Well, I'll answer that. If you have 13 minor deficiencies and DFR refuses to receive you for too many minor deficiencies, DFR, if you request a reconsideration and we determine that the number of minor deficiencies is now 9 or less, we will rescind the RTR, but the expectation is that the applicant has to address all the minor deficiencies, adequately address all the minor deficiencies. Now, if those minor deficiencies are then adequately addressed, then you will receive a filing acknowledgement letter with the original receipt date.
Thank you.
You're welcome.
Morning. This is Sharif Ahmed. I guess this question is for Dr. Ren. I know you presented in one of the slides some of the acceptance criteria. So for typical bias studies, the regulatory requirement is for the confidence interval is 80 to 120%. And I know in terms of how FDA interprets and applies that and communicated that through guidances is that that would be 80.00 to 125.00%. As long as that the range is within that, it would be considered acceptable. We have seen deficiencies where for replica design studies where the upper bound of confidence, I'm not a biostatistician, so please, if I mess up the terms, forgive me, but I guess the question is still important. So if the limit is less than 0, we have seen requests for new studies when the limit is 0.001. So the question is, what is the basis of that interpretation? Is there any regulatory basis, or what is the practice, and what is the, when should we know that it is failing or passing until we, or should we wait until we find out from the deficiency letter?
Can we get her lapel back up? Thanks.
For the general BE criteria, the B limit is 80 to 125%. If you choose, like you mentioned, use the 4-way crossover, I'm assuming they try to estimate the variability of the test at the reference. Maybe they want to use the reference scale approach. If the variability is greater than 0.294, we will allow to use the reference scale approach. If the variability is less than 0.294, the average BE will be used. So the BE criteria will be 80 to 125%. If the higher than 0.294, the reference scale will be used. The BE criteria will be the two limits. One is the upper bound for the OPK parameter should be less than 0. The rationale why this 0 come from, when you look at the progesterone capsule guidance, we have the rationale, how we divide it, how we come up with the 0. And also the point estimate has to be within the 0.8 to 1.25. Both criteria, if they met, then we can conclude the test and reference are bioequivalent.
Okay.
I'm sorry, I don't think that answered my question, so maybe I can clarify.
Yeah, if you can ask it briefly and succinctly, yes, please go ahead.
Yeah, so I guess the key part of the question is, what is considered less than 0? So if the result comes out to be 0.001, is it considered more than 0? Or if it comes out to be 0.00 to the 10th, 1 at the 10th decimal, is it still considered more than 0?
So would it be fair to say that the succinct version is what is less than 0?
Yes.
Okay.
It's still considered greater than 0. So we're not allowed to round up, round off. If the calculation got the 0.0001, we still consider greater than 0.
Okay. Online question for me, please.
Sure. This question is for Varun. If an applicant is not using a current FDA 356H and is using the previous version, will the ANDA be RTR'd?
Right. Thank you for the question. So just to make sure that as long as the form FDA 356H is the most up-to-date form and not expired at the time of filing, that would be accepted.
Okay. Microphone number 2 up close. You can tilt that down a little bit if you like.
Hi, this is for the a question for the ANDA reviewers. So there are times on the RLD label where an excipient has a specific level specified. You know, like maybe glycerin is X%. Is the expectation for a generic to have an assay test upon release to support the label having the specific excipient quantity there?
That question is actually outside the scope of what DFR would look for. That's more of a technical review. So maybe later on down the road, they might require that of you, but at the DFR standard level, we would not.
So it wouldn't be a deficiency.
Microphone number 1, please.
Hi, good morning, panel. My question is for Aiden. With relation to the justification of impurities table that you discussed, so for justifying the limits for unspecified impurities, would the agency typically still consider references to monographs such as USP or EP as a valid scientific literature?
You said which type of impurities you were talking about?
Typically specified impurities, which are sometimes not there in the, say, USP monograph, but they are somewhere in the EP monograph or a JP.
You are saying unspecified, right?
Yeah, sorry, unspecified impurities.
So if you look at I refer you to the impurities guidance. The impurities guidance explains that for unspecified impurities, there is your proposed AC should not be greater than the regulatory IT threshold. So in the impurities guidance, it does state that DFR will refuse to receive you if you use a monograph to justify for an unspecified impurity.
Okay, thank you.
Take an online question, please.
Okay, this is for Captain Doan. For a CBE 0 can you implement it immediately or must you wait 30 days before implementing?
Okay, so the question is, how long do you have to wait to implement for CB0? And the answer is immediately. So for CB0, you can implement it immediately. CB30, you have to wait after 30 days.
Microphone number 2, please.
Okay, once again for the reviewers, Aiden, Varun, and Julie, not sure which one, but I just want to make sure that I understand the big picture of how the items are counted and arrived at and what happens subsequently. While I certainly don't expect the agency to sit around and review a file that has 800,000 items and list each one of them individually, I just wanted to know, is there a point at which a predetermined number of items at which the file review would stop and you said, let's just send this back now? Or if it's a reasonable amount of items, let's say 18, 15, 11, is an entire review of the file done, and then knowing that these are the only items that need to be addressed, if you have to go back to square one, is it safe to assume that that's the way that the process works?
So for your question, so DFR will review the entire ANDA once it does make it into our division. So there is no number where we will say, well, this ANDA should go back and have the reviewer, have the applicant fix it before we finish the review. So we do review it entirely.
Another online, and if there's questions here in the room, please come on up to the microphones. Another online question, please.
Okay, can you please clarify regarding the requirements for the 314D patent verification? We've been told previously that a statement in the cover letter is not sufficient and that a signed cert must also be included in the patent section.
That is something that is reviewed by the policy group. But my understanding is that science certification that you're referring to is a requirement at the very first time. So after that, I'm not really sure, but you can email the regulations.gov email and verify with them.
Microphone number 2, nice and close for me, please.
So we have one approved ANDA where I conducted tox study, and we have given justification for that impurity as a tox study. So that ANDA is commercial. Now I am going to submit another ANDA, and it contains same maximum daily dose, same active ingredient. And now can I give a reference of that ANDA for tox study for justifying that non-impurity?
So you're asking if you can use the level of an inactive ingredient that was in an approved ANDA to support an amount of an inactive ingredient in a proposed new ANDA?
No. I am asking about the one of the non-impurity we have justified label based on the tox study.
Okay.
And that ANDA is commercialized. Now I am going to submit another ANDA which contains same active ingredient and maximum daily dose. So in order to reduce the review cycle, can I give a reference of that approved ANDA?
So you're saying for the level of impurity?
Yeah.
Okay, so you're using you have an approved ANDA with an impurity at a level that was approved by the agency. And you are proposing a drug product that has an impurity level at the same amount as the approved ANDA. So from a filing perspective, you would want to provide that information as justification or to support the amount of the impurity level.
Yeah.
From a filing perspective, that would be acceptable. However, we can't make a comment to how the review disciplines will look at it. But from a filing perspective, if you provide that as support, then we would accept it.
Online question, please.
Okay. If a BE study is conducted on an exhibit and the exhibit batch fails, and the study is repeated using the same exhibit batch with a different study design, Is it required for an applicant to submit the complete failed BE study, or is a summary table sufficient?
So if they use the same batch and one study failed, passed, the applicant will need to submit the at least 16 summary BE table for the failed study and briefly the clinical study report. In the summary, the applicant will need to submit the rationale why the study failed, the possible rationale. During our review process, we will evaluate and we may request for a study report, analytical report, and SAS data set to verify the reason of failure.
Microphone number 2, come up nice and close for me, please. If there's any other questions in the room, feel free to come up with the mics.
This question is related to retention samples. So actually guidance mentions that we have to retain bioequivalence, retention sample for test and as well as reference products. The quantity mentioned in the guidelines is at least 5 times the quantity required for perform all the release texts. But there is no specific number mentioned. However, the higher threshold is 300. I understand that, but what is the reason for not mentioning the minimum number so required for retention samples? So that, why I'm asking this question is, in certain cases, the cost of the RLD is very high. So during that time, the sponsor is in a question in that whether I should go for minimum requirement as a 5 times the quantity required for release text, that is actually unknown and there is an uncertainty behind it. But if I go for a 300, then the costing will be very high. So in that scenario, I think if there is a mention of a minimum requirement in the guideline, that would be great. So what is the thought process in it?
So this depends on dosage form. So whether it's complex or just a solid oral dosage form. Current agency is working on the map to classify how many minimum units should be returned. We're still working on.
Okay, thank you.
Okay, next one, nice close to the microphone, please.
Good morning. USP monograph has a TLC method for estimation of impurities. Is the TLC methods are acceptable to monitor the stability studies actually?
Can you repeat your question?
USP monograph has a TLC method for estimating the impurities actually.
USP monograph has what?
To estimate the impurities. And is the TLC methods are acceptable for stability study monitoring?
Are you asking if the TLC method is acceptable?
For stability monitoring.
For stability monitoring?
Yes.
That's outside the scope of our review. That would be a question that you would want to ask OPQ or the chemistry reviewers. Yeah, there's a stability presentation after this, and there's actually a presentation regarding impurities. So that would be a question you would want to direct to them.
Okay.
All right, let's take an online question, please.
Okay. Is submitting RLD updates to their labeling considered an unsolicited amendment?
Question is, are RLD update submissions considered an unsolicited amendment? Yes, generally speaking, yes, it is.
All right, microphone number 2, up nice and close for me, please.
Question to Division of Filing Review. For a limit of unspecified impurity in the USP monograph, suppose we are providing a justification based on it, suppose it's a metabolite or something. How do you take the justification? It is RTR straight away, or it will be accepted and then reviewed for the chemistry?
I only heard the first part of your question. So for an unspecified impurity that was in the USP.
It is not in the USP.
It is not in the USP. Okay.
I'm providing a justification for the limit, a scientific justification.
So with anything that's written in a guidance, those are recommendations set forth by the agency to meet the requirements set forth in the regulations. So if you as an applicant provide a justification for an unspecified impurity, that's a scientific justification, and you provide information within your original ANDA submission, then it would be accepted for filing, but then that would be a potential deficiency that may or may not come up during the technical review.
Because there is not a clear definition of the scientific justification. So as a division of filing review, maybe you will not be having that resources or capability or technical expertise to decide whether it is acceptable or not. So you are going to accept that one and then forward it to the next level of review?
So if you provide a justification for your unspecified impurity and why the proposed AC is greater than the regulatory IT threshold and it's a scientific justification, from a filing perspective, it would be acceptable.
Microphone number 1, nice and close for me, please.
Hello, good morning. I have a question regarding the 356H and the section 32P31. If I update the 356H, just to make sure that the facilities are including are only for commercial manufacturing and not for exhibit batches, would that extend my GDUFA goal date if it is just to make sure that what I'm including, I'm not adding new facilities, just only including what is for commercial manufacturing?
Just to make sure I have your question correctly, you said if you want to update your 356H, just to clarify which sites are doing the exhibit batches?
Just for commercial manufacturing.
Just for
the
commercial?
exhibit batches.
Okay. So again, that just depends on what might have been understood or submitted in the original submission. And of course, again, that's an evaluation that's done by OBQ and the inspection team. So it just depends on how it was interpreted the first time.
So if you update your 32P31 just to separate your commercial batches in one table and your exhibit batches in a different table, and those commercial batches are identical to the ones that you're proposing on your 356H, would that affect your GDUFA goal date?
I couldn't hear the beginning part. You said?
If you're updating your 32P31 just to separate in one table your commercial manufacturing batches you proposed, and in a separate table, the exhibit batches, instead of having everything in one table, your commercial batches will be identical to the ones on your 356H. But there are no new facilities being added. It's just for clarification to separate commercial from exhibit. Would that affect your GDUFA goal date?
Yeah, again, I mean, I just go back to exactly how originally it might have been interpreted when you first initially submitted the data and the information. And that's an evaluation, like I said, is done not by us, but rather the OPQ team and the inspection team.
Okay, great. Thank you.
Microphone number 2, please.
This has reference to RLD update. I asked this question yesterday. Probably I'm repeating the same thing. And since it's applicant's responsibility to update in line with the RLD label for a pending ANDA particularly, when we submit the labeling amendment, why does the goal date get affected?
So your question is that when you update the labeling or you submit updated labeling information, why does it affect the goal date?
Yeah.
Okay. So the answer is because it is an unsolicited information, and that's why it's a big part of why I had that in the presentation. Because we do see a lot of times the updates are there and they have been there, and applicants are not monitoring the updates until the last 30 days prior to the goal date. And so, that's a big reason why we mentioned it today. But it is an unsolicited, and unsolicited amendments do extend the goal dates.
So what would be your advice? Should we wait for the agency to communicate to the ANDA holder? Because when we volunteer, because we do monitor it regularly to avoid affecting the goal date, we volunteer, we update the label before the agency communicates to us. So when we get the acknowledgement, we realize that the goal date has been changed. So how do we prevent the labeling update from affecting the goal date?
Well, I mean, at the end of the day, you're not going to be able to market or get approval for your product without that update. So, I mean, there's no way around it. So the best thing is just you monitor, and once you see the update, you submit that update as soon as you can. Okay.
Let's take an online question, please.
This question is for Aiden. On the Q&A on stability testing, question 4, section C, page 6, it indicates that a minimum of 2 lots of the drug substance should be used to prepare the 3 primary batches of the drug product. Could you please clarify what is meant by primary batch? Is that 2 lots per strength or 2 lots per application?
So per the stability guidance, it's recommended that the applicant provides in 2 API lots per strength out of the 3 batches that's used per strength.
Microphone number 1, nice close for me, please.
Hello, this is a question for about the terms for the justification table. I just noticed that you used the term as the unspecified impurities, but according to one of the IRs we received from the agency, we are required to change the terms to unspecified degradation products. So just wondering if that will cause any deficiency.
So your question is regarding a specific ANDA, and it's outside of the scope of this presentation. Our understanding is that an applicant should be submitting the impurities tables based upon the recommended format that's on the agency's site. So it should have specified, identified, specified, unidentified, and then unspecified impurity.
Sorry, the question is about the, should we use the impurities or degradation products? And also the degradation products, is that, shall we must use these terms?
So, like I said, you should follow the format. So if you look at the tables on the agency's site, it identifies how you should format your tables along with the titles to said tables.
So just to follow the forms.
Yeah, I would use the recommended tables that are on the agency's site.
Thank you.
Okay, we got time for one more online and one more in the room. Let's take an online, please.
Okay. For Module 2, what sections require both the PDF and the Word documents? Can you clarify?
Sure. So in Module 2, you want to have in Module 2.3 for the drug substance, and again in Module 2.3 for the drug product, they both require PDF and Word. And as far as Module 2.7, the summary data tables as well require both PDF and Word. Thank you.
Okay, and right here, microphone 2.
Yeah, this is in continuation of the question on impurities, unspecified impurity, the ICH versus or the regulatory specification as against the USP. And we have observed if ICH says not more than 0.2 and USP says 0.24, are they different?
Yes, they're different because 0.2 is different from 0.24. However, that question might be better answered by the panelist that is coming up after us.
Sure. And we all understand that USP monograph is based on agencies approved NDA or ANDA.
I don't know that I'm understanding the question. What's the question?
No, the USP monograph. Okay, the USP publishes a monograph based on approved specification by the agency, either based on NDA or ANDA.
Okay.
So that means it's been approved and accepted by the agency. So I understand that I will ask this question probably in the impurity section, but just wanted to understand why there is a difference in the approach.
That would be a question you would have to ask the panelists after us.
Okay. And we will sneak in one more question before we go to the break, if you can make it real brief and focused.
So there is a RLD in Orange Book which is discontinued, and we have reference standard is in a Orange Book. Both are different. Now my question is, RLD is having totally different inactive ingredient, and RS is having totally different inactive ingredient. Now I am going to formulate my drug, and it will be practically with a reference standard. So do I need to justify the difference of inactive ingredient in my anda?
Can you, so you're asking, the inactive ingredients are different between the discontinued RLD and the reference standard?
Yeah, because my basis of submission is RLD. So when, yeah, please.
So it depends on the dosage form. So you are under the regulations pursuant to 21 CFR 314.94A9. You are required to be Q1, Q2 same to the reference listed drug. And this is outlined in the guidance for industry on reference listed drug that I believe is still in draft. I don't know if it's in draft or if it's finalized, but from a regulatory perspective, you must be Q1, Q2 same to the reference listed drug.
PSC guidance is available for that product. So I have conducted BE study as per the PSC guidance, and then I found that my drug is equivalent to reference standard.
So for base of submission has to be the RRD.
Yeah, RRD.
If the drug product, if this is solid dosage form, not injectable, it's not through the waiver request. Then the B study in vivo B study has to be conducted, use reference standard.
Yeah,
and I'm going to cut off just to keep us on schedule. This is sounding like a very specific instance, and we will have a slightly longer break because our panelists and our presenters this morning were nice enough to shorten it up so you can catch them during the break. Please help me thank our panel for sharing their wisdom. Because we did